Hello and welcome to my channel Pharmacy in Depth, where we'll talk about pharmacy related topics in detail. In this video we are going to talk about immunity and its types. So let's start the video. So immunity is the resistance exhibited by the host against any foreign antigen and towards injury caused by microorganisms and their products. Antigens are molecules capable of stimulating body's immune response. Immunity is divided into two types: innate, which is present at birth, and acquired or adaptive, which we develop as we grow up. Innate is divided into specific and non-specific, and acquired is divided into passive and active, which are further divided into natural and artificial immunity types. The essential components of innate immunity are epithelial barrier and cells like dendritic cells, macrophages, neutrophils, and it protects the body during the initial hour of the infection. Whereas components of adaptive immunity are basically B lymphocytes and T lymphocytes and protects the body during the later days of the infection. Now let's see each immunity in detail. First is the innate immunity, which is also known as native immunity. It's resistance to infection that an individual possesses by virtue of his genetic and constitutional makeup, and it is not affected by prior contact with the microorganisms. It is further of two types: specific and non-specific. Specific is particular to specific pathogens, and a non-specific provides resistance to infections in general. There are three levels of innate immunity. First one is the species immunity, which is the immunity shown by all the members of a species. For example, birds are immune to tetanus. Second is the racial immunity, in which a particular race of a species are immune. For example, Negroids in US are more prone to tuberculosis than Caucasians. And the third is the individual immunity, which is specific for each and every individual of a species. Now let's have a look at the defense barriers of our body. We have anatomical barrier, phagocytic barrier, blood proteins and cytokines. And the first line of defense of our body are saliva, tears, skin, mucus, stomach acid and microflora of the gut. Now let's have a look at all the anatomical barriers of our body. First one is the skin, which acts as a mechanical barrier and provides bactericidal secretions, and resident skin microflora prevents colonization by pathogenic microbes. Second one is the respiratory tract, where the inhaled particles are arrested in the nasal passage on the moist mucous membrane surface, which has hair-like cilia that propels the particles towards pharynx, where it is swallowed or either coughed out. The third one is the intestinal tract. The mouth possesses saliva that has lysozymes which kills microbes. The acidic pH of the juices kills swallowed microbes, and the normal microflora of the gut prevents the colonization by pathogenic microbes. The next one is the conjunctiva, where the tear flushes away microbes and all the dust particles, and lysozymes in tears has bactericidal action. The fifth barrier is the genitourinary tract, where the urine eliminates bacteria from the urethra by its flushing action. Acidic pH of vaginal secretion makes it free from microbes, and in males, semen is believed to have some antibacterial action. The second barrier is the phagocytic barrier. Phagocytosis is a type of endocytosis where a cell engulfs foreign microbes and particles, and it is the major mechanism to remove pathogenic bacteria from the body. We have professional phagocytes like neutrophils and monocytes in blood, macrophages and monocytes in bone marrow, etc., which have a receptor on their surface, and phagocytosis is the primary function. And we have non-professional phagocytes like lymphocytes in blood and lymph nodes, epithelial cells in skin, which do not have a receptor on their surface. Here are different steps involved in phagocytosis. The first step is chemotaxis and adherence of the microbe to the phagocyte. The phagocyte then ingests the microbe to form a phagosome. This phagosome fuses with the lysosome to form a phagolysosome. These enzymes in the lysosome digest the microbes to form a residual body which contains the indigestible material, which is then discharged out of the phagocyte in the final step. The second type of immunity is the acquired immunity, which is the resistance developed by an individual during his life by recognizing and selectively eliminating foreign molecules or microbes. It provides second line of defense against infections. It is of two types, active and passive. In active acquired immunity, resistance is developed as a result of an antigenic stimulus, and in passive acquired immunity, resistance is transmitted passively to a person in a ready-made form. Here is a difference between active and passive immunity. Active immunity is produced actively by the host's immune system, whereas passive immunity is received passively and there is no active participation of the host's immune system. Active immunity is induced by infection or by immunogens, whereas passive immunity is conferred by administration of ready-made antibodies. Active immunity is long-lasting and effective protection, whereas passive immunity is short-term and provides less effective protection. There is a lag phase in active immunity, whereas there is no lag phase in passive immunity. Immunological memory is present in active immunity, whereas there is no memory present in passive immunity. Booster effect is seen on subsequent dose in active immunity, whereas it is not seen or less effectively seen in passive immunity. Negative phase may occur in active immunity, whereas no negative phase is there in passive immunity. Active immunity is not applicable in immunodeficient patients, whereas passive immunity is of much help in immunodeficient patients. Active immunity is of two types: natural active immunity, which is developed as a result of clinical or inapparent infection. For example, a person recovered from measles will have antibodies of measles. They are long-lasting immunity but depends on the pathogen type. And the second one is the artificial active immunity, which is the resistance by vaccines. Vaccines are preparations of live or killed microbes and their products. Similarly, passive immunity is also of two types: natural passive immunity, which is a resistance transferred from mother to fetus or infants through placenta and milk, and artificial passive immunity, which is the resistance transferred to the recipient by administration of antibodies, for example, in case of hyperimmune sera of animal or human origin and convalescent sera. Now let's see the mechanism involved in acquired immunity. The first one is the cell-mediated immune response, which involves T lymphocyte and eliminates intracellular microbes that survive within the phagocyte or infected cells. And the second one is humoral immune response, which involves B lymphocyte and is mediated by antibodies and eliminates extracellular microbes and their toxins. What happens in cell-mediated immunity is that T cells are found circulating in blood, and when they encounter the antigen-presenting cell, which can be a macrophage or an infected cell, it forms a major histocompatibility or MHC complex. Once a T cell encounters an antigen, they proliferate in presence of cytokines to form cytotoxic T cells. The cytotoxic T cells destroys the infected cells by inducing apoptosis. T helper cells stimulates plasma B cells to produce antibodies. And in case of humoral immunity, antibodies are produced and secreted by plasma B cells, which are activated by T helper cells. B lymphocyte recognizes a specific antigen. It proliferates and differentiates into antibody-secreting plasma cells. These antibodies bind to specific antigens on microbes and destroys them. So guys, this was all about immunity. Thanks for watching. I really hope you liked my video, and if you did, like it, share it and subscribe to my channel.